Thank you so much. I just wanted to thank the all investigators and clinicians for their wonderful update on this exciting therapies for these devastating diseases. I did made a slide for some potential questions that can break the ice, but I want to first open up to the audience and as well as the people in online to see if they have any questions for our people. Yes.
Laura, for AGS for baricitinib. So I guess we've treated some patients with baricitinib, and I guess mentally in my mind, I was carrying around the model that baricitinib is an immunosuppressant / modulator, and so if they get sick, you should be kind of freaked out and thinking about stopping it. But then you said the opposite. Could you clarify?
Correct. I'm saying the opposite. When you look at how we're modulating the immune system, we're actually just bringing it down closer to normal, right? And so we're taking an immune system that's hyperactive and turning it low. If you look at interferon scores on baricitinib in the context of infection or vaccination, they actually go up, but up into the normal range. And so we work with our immunocompromised ID team to say, hey, do you think this is a funny opportunistic infection that would warrant stopping it? I will name, we have pushed through in every single case with the exception of one. We had one child that in retrospect had persistent infections that were opportunistic. They were also on chronic steroids at the time. So I'm very wary of dual immunosuppressant therapy causing much more of an immunocompromised state than baricitinib, which I call much more immunomodulatory. These kids, we follow their immune function very intensely. We also follow viral titers too. So I'm looking for herpes viruses. Those are the ones we think would cause the most drama. I'm looking for BK virus, which we know, at least in the immune world and the transplant world, is something we need to follow. So that being said, I follow a lot of stuff. I have not done a lot of dose modification for that. I think the one reason I have come off of baricitinib is persistent anemias or thrombocytopenias, which can also be driven by disease. But I've had a couple kids with really, really refractory, and I have switched to anifrolumab in those very rare cases. The NIH protocol actually does dual therapy with anifrolumab and baricitinib. That makes me really worried hitting the immune system in two ways. So we have transitioned everyone fully onto just anifrolumab with good success.
Thank you.
Can I say one more thing really quickly? I think immunosuppressant medications makes neurologists really nervous. We have had to lean into this space because we have really failed to find rheumatology and immunology teams that felt as comfortable with AGS. So I actually do all of my own prescribing with anifrolumab and baricitinib at this point, which sounds crazy, but it was much easier for me to learn to be comfortable with a small number of drugs and their monitoring in partnership with support teams rather than trying to convince other people to care about AGS as much as we did.
Thank you. We have one question here.
My question's for Stephanie. I'm excited to see the data coming out on both of the ASO trials that you talked about today. And my question for you is regarding the placebo control arm for the Alexander disease. You know, for most of our rare disease clinical trials, we don't have a placebo arm, which is, you know, obviously a common criticism of clinical trial design. And for that trial, they do have a placebo control arm. And my question is really the risk benefit and the ethics of making that decision, because you are having kids undergo an LP. So it's not just, you know, like a blood draw or something. There is an additional risk to that placebo control group. Can you discuss at all the decision to have the placebo control in this trial?
I don't know exactly because I didn't develop it. The families were okay with it. I think because there weren't any other options for treatment. And, you know, they are gaining other biomarkers and things when they're doing the LPs. So it was necessary to get CSF even in the groups that aren't necessarily getting the drug. But there was in the course of the trial. That's why they changed over to the sham group. I think it was from FDA kind of comment that maybe we shouldn't be giving them this placebo injected into the CSF space. So they went to more of the sham, which created just a little more complication with the study because now you had to have this other group of investigators to that knew whether or not the patient was actually getting the drug. But you know, when you have these rare disease groups and you don't have as much, you know, information on them long term. I mean, Amy Waldman did a lot of good studies about getting CSF samples for the natural history, but when you have these really slow diseases, it's really hard to see changes. And so I think collecting as much information as you can is just important to see if there's actually any change.
Thank you. I'm sorry, I have a follow-up question that maybe.
Oh,
Go.
I was just going to say that everyone in the trial also gets on study drug. There's an open label period at the end. So even those who are on placebo do get access.
Yeah, I was gonna say, because I mean, that was important for the families to participate, was that knowing that they were going to get drug at some point, even if they weren't getting it initially. There also was, I can't remember how many weeks it was. So, you know, as the investigators, we were blinded, but main study people were monitoring that. And if they saw that there was somebody on placebo that was declining, they would switch them over to drug without us knowing. We'd be blinded to that, but that was part of the protocol. Yes, yeah.
I kind of have a follow-up question to that, and that's come up with as I watching all the listening to all the investigators, is that how do we involve in this context? How do we involve the patient groups and families so that we can get better research goals and improve clinical trials? And this is for all of our panelists. I know it's a lot of question, but I know that the sort of the question that I was asking alludes to that. It's like what feedback can we get from our patients that can improve? How do we actually translate these therapies to be more effective and applicable to all the families that need them?
But I'll just say that as part of the results that they're collecting, they do collect a lot of most bothersome symptom questionnaires for the families to make sure that they're kind of hopefully addressing with the therapy what's really bothersome to the families.
I'll say in my experience so far with the adult community, for ALSP in particular is a very active community that's involved in the advocacy group, does a great job of coordinating input with surveys. I mean, I think the limitation right now is funding to be able to get access to all the different communities to find out that their feedback of what they're comfortable with versus not comfortable with, even for treatment outcomes and what they're willing to accept as adverse outcomes. And we're learning, but being able to have access to the community is obviously the key.
At every single step of the way. I mean, they help us with the trial design. They help us choose endpoints. They help us to decide what's really important. So, yeah, I think it's a partnership starting ideally from the very, very beginning.
Yeah, I think that's incredibly important. And I think the point that not just understanding, you know, outcomes and what's important to families, but also the level of risk they're willing to tolerate, because sometimes that's very different than the level of risk that we as clinicians and investigators are comfortable with, and being able to grapple with that a little bit. Also at the level of designing trials to see like what is actually feasible for families to go through and what would they be willing to do.
I think one of the good experiences I've had over the past couple of years has been with for the VWM consortium pulled together maybe 8 or 10 people across the world who took care of patients with VWM, and recognizing that that the field was kind of on the brink of starting therapies, that we needed the kind of expert advice of clinicians taking care of patients, but then recognizing that there's a limited number of patients with the disease, and if you're going to stagger clinical trials, like making sure you don't allow one company to kind of swoop in and get all the patients on a trial that didn't make sense, or making sure that if you collect a natural history, that it didn't end up locked away in the vaults of some drug companies. So I think that the professional community has a kind of hand-in-hand role to work with the patients to achieve kind of the goal that you're all working for. And so I think that although we I think it's what the families and patients need and want should there's an important role for the clinical and professional community to kind of be the partners to make those goals happen. So I think we shouldn't be afraid to kind of provide that professional level of advice.
Thank you. I think there was one more question, last question.
Yeah, I have a newborn screening related question for Dr. Adang about when newborn screening for adrenoleukodystrophy started, there were these anecdotal reports that patients with AGS were identified. But I guess that is not a marker that you're pursuing, or why did they have lysophosphorylcholines elevated?
Yeah, so we do get a very small percentage of our AGS cohort is identified incidentally through the ALD newborn screening. It's just not everyone. I think they're getting secondary incidental peroxisomal dysfunction, and it's not a primary peroxisomal disorder. We've collected, oh, forgive me, I think around 70 newborn screening cards of the children with AGS. And so we're working with Joe Orsini, if he's still here, and Mike Gelb to try and come up with an assay that's sensitive and specific and helps us to discriminate from congenital infection. I think that that's the big place where I'm really worried. I think the other place that as we're getting better therapies in line for newborn screening readiness, I'm also worried about discriminating and stratifying subtype and severity because there is a really broad phenotypic spectrum, and I wouldn't want to start an immunosuppressant at birth on someone that might be destined to have just mild ankle spasticity later in life, whereas some children are absolutely devastated by this disease. And so we're doing some early markers and have a paper that we're submitting. Hopefully it was submitted while I was here on some biomarkers that might help us with some stratification for risk, because I don't think everyone that has a change in the AGS gene should get immunosuppression for lifelong. I think that that's a really big ask of families. I think we're also looking much more towards in the future having targeted therapies that are one-time delivery rather than this kind of like chronic immunosuppression need too.
Question here. Doctor.
I just wanted to say, and I stepped out for a second, so maybe I missed it, but I think with the question around having the placebo arm, it's really important to remember some of the history, because the first big leukodystrophy trial was Lorenzo's oil. And after decades, there was no clear answer because there wasn't a placebo arm, because maybe we listened too much to what we thought patients wanted, and then the result was that we didn't end with an answer. So I think it's important we remember some of the past failures and why those questions weren't able to be answered.
We have a question. Jen.
Thank you all for this talk. Just wanted to ask what we maybe can do as clinicians with a lot of these very rare diseases and the kind of trials going on. They're very specific centers. And I worry a lot about access for our patients with language barriers or financial barriers. So just wondering what we as clinicians or even organizations can do to help with some of those issues, because I see that with some families being incredibly well resourced and able to go anywhere for these trials and others not able.
I can just say that I know with the PMD trial, the assessments take a long time. It's like 4 days that the patients are there for all of their assessments and then getting their drug administration. And again, sometimes there are overnight stays afterwards and assessments the following day. So it's a big ask to do that every 12 weeks and then have clinic visits in between. So that has definitely been an issue for some families. And I do worry that it's only going to be those families who have these jobs where they can work from home or have a lot of resources that they can do that. The companies pay for travel, which is nice, and they pay for all their meals and housing when they come. But again, being able to leave your job for 4 days every few weeks, and if you have a disabled child, then that's both parents are having to travel for that. So it's a big ask. And we have, and the kids that are at my site are not from my state, so they're all traveling. So I mean, it'd be nice if there could be a site at every place, but unfortunately, that's not going to be possible. So the kids are going to have to travel. Yeah, I always worry that our research is kind of slanted in a certain way. But I think that's true probably of all research. I don't know a great way other than organizations maybe being able to support families being able to travel for these trials. Again, I think the companies try to support it as much as they can.
Sorry to interrupt. I just had a question about is there something like an expert directory where us as clinicians can sort of contact investigators that are running these trials to facilitate the communication? Is there room for that, or has that been proposed or done? Alluding to the issue of access and barriers, because these are all too rare diseases that we may just see one every few years, but we want to offer the best possible care for them. So I don't know, I'm sorry for interrupting, but it occurred to me that question.
Well, I think all the site PIs are listed on clinicaltrials.gov, so you can just reach out to that and also Google and see who's written papers in this space. We see so many kids with AGS at CHOP. I think we're up to 200-ish at this point. And I don't want to see all the families with AGS in the country. I don't, I promise. So we've done webinars. We've posted them free. If you pay a little bit, you can get CMA credit for it. We've created brochures. We share our letter of medical necessity for baricitinib. Like, we literally have a packet where a new doctor reaches out to me. I say, thank you so much. If the family can travel, we'll see them. If not, here's all the information you need to do this on your own. And so we have a lot of families that see us once, and then we have something we call a shared care model, where we meet regularly with the local teams. We provide information, we do oversight and guidance, but like, we try and empower local teams, because I think AGS is probably going to be the second most common leukodystrophy after ALD. And so there are a lot of undiagnosed families out there that need to get local care just for that same reason of equity and access.
If there's no other questions, I do have one. We've been hearing about all these wonderful potential therapies, but I always question whether there is a faster, better way to translation. Is there a need to change the way we think about translation in the context of these rare diseases? Can we come up with a better way to provide help for these families? Is there some new idea, some new way? Is that even the correct question to ask? I mean, is there another method? But anyway, just wanted to get your thoughts on that.
I guess I'm moderately optimistic that yes, there will be some better pathway. I think we don't know what it is. But I think we've kind of, we're kind of going through options, right? So I think there's been a kind of a run to try the repurposed drug route. I'm currently not I don't think that's going to be the success. I think that if it looks like the gene therapy AV9 process, at least currently, is very expensive and difficult. So that's probably not going to be the win, except for like, more common rare diseases. But maybe, I think ASOs are proving to have like their own set of challenges, but maybe there's some therapy down the line that we just haven't hit yet. And when we hit that, it'll be kind of a modular, interchangeable thing, or maybe there's like downstream steps, like for AGS, if they all have the same common pathway, maybe there's some drug that you can like click that same common pathway. So I think we don't know what it is, but I think, I don't think that's a reason to, I think we should be hopeful.
Hi. I kind of don't want to talk about it, but I will. The gene editing case that came out of CHOP baby KJ that hit the news. That's my partner, Becca Ahrens and Karen Missionero. They did a beautiful job, but what hit the news wasn't actually what's cool. It's not the fact that it was gene editing for a single patient. It was the fact that they got both the NIH and the FDA on board to make it a basket trial. They built the trial, they got approval, they were ready and waiting for the perfect sequence, and therefore created a fit for purpose gene editing with all the regulatory infrastructure set and ready to now flip into the next patient. So it's an N of 1 + 1 + 1 + 1, which the news didn't hit at all. But like, I think it's stuff like that is going to be really helpful. Thinking about what can we take from one specific instance, get that regulatory approval. And now when they have another baby with the same general disease, they can just flip out the cassette and do some very small testing of just the cassette that's specific to the gene because everything else was standardized. And so I think that idea of creating a regulatory platform to be able to flip into a space where we're sharing resources and that regulatory burden, I think will shave a lot of time off. And so I was really sad, and I will name the team was really sad too, that what got picked up was this bespoke therapy. It actually wasn't a bespoke therapy. It was a therapy waiting for the baby, right? It wasn't a family that reached out to them and said, please help. It was a therapy that was ready to be able to be rapidly mobilized in a way we haven't seen in clinical trials before. Yeah, I agree. It's much, much cheaper than where we can share resources. I think coming up with central repositories of data where we make regulatory data that is ready. So within the GLIA CTN at CHOP, we say if a drug company wants to see wants us to partner with them, any natural history data and biomarker data that's not proprietary to the company has to be fed back into the natural history study. And so I have projects where Biogen wanted to look at samples of patients with AGS and they ran up panels. Now the data is back in GLIA CTN. It is now a shareable resource for the community. And so I think it's thinking forward to not siloing information, but saying, if you want to be in this space, we're going to just share. That's just the way we're going to do it.
My insight is not as amazing as what they did, but just on the adult clinic side, I've been thinking of like, what are all the things that I can collect in real time in a clinic visit that would be an outcome, so that we already have a comparator even for the individual, and sort of developing standardized ways to collect that and have insurance pay for it, and to follow over time. And so that's sort of something that hoping to expand, that we're basically already doing the standard outcomes of a clinical trial.
Hi, thanks for all those talks. So I have a question. I think it's probably mostly directed for Amanda and Laura, but thinking about outcome measures, that you're following during the gene therapy trials, how you're comparing that to natural history, the patient's previous trajectory, what you can get, especially if you have to, if you're using the option of remote assessments. And I had noticed, I think, so Amanda, when you showed for the Canavan disease trial, that there were several scales that I recognized for like infant motor development, for instance, that you got remotely, which is really exciting. But then when you showed like that nice heat map, there were like gross developmental milestones identified. And I was just sort of curious, first of all, like your thought process with selecting those outcomes, and how you sort of choose maybe some things that are more simple but more accessible versus the scales. And I think sort of similarly, I'll just tag this on for Laura as well, for the AGS paper, I think that came out a few years ago. You showed developmental skills and there was like a model that you used. So I'm just sort of curious if you all are leaning more towards these specialized scales, which have their challenges, and you need people who know how to administer them and they're in certain context, versus looking at, I guess more gross milestones that you can really get everywhere. Thanks.
Yeah, I think that's a really excellent question and something that we've struggled with a lot. I know particularly in the Canavan trial. And the reason is that kind of at baseline in the natural history of the disease, these kids are at floor on many of these scales. And it's really difficult to pick up subtle differences or subtle changes over time. I think in our higher dose cohort, we're seeing maybe more robust change in the motor outcomes, but really in the low dose cohort, I can say from experience, kind of seeing these kids in clinic over time, you see differences in them, and the families see differences, and they're not necessarily captured in any of the scales that we're using. And so while, CDC milestones is one of the things we looked at, and, GMFM, and Bailey and all of those scales, I would say a lot of the scales, it was really hard to find appreciable amounts of change. So, in that sense, we've worked very closely with our physical therapists in particular to understand, like, what are they seeing when they're doing some of these assessments and what might actually be meaningful change. I will say there was a lot of work done to develop a Canavan disease specific rating scale, looking at kind of domains that are important in Canavan disease, which is fantastic in that it captures things that are very important, but I think is also very difficult to see change on because it's very kind of a crude scale. So it is something we struggle with, and I think the way we've managed it is to have very consistent raters between the patients in the natural history and in our trial, and have started to look more carefully at some of these very subtle changes, like subtle changes in head control, in posture, in rolling, reaching, those sorts of early skills.
Yes, I agree with all of that. You just have to, I mean, I think one of the challenges we have is floor effect with really outcome measures where our children have functional skills that just aren't captured by clinical outcome assessments that were developed for cerebral palsy, right? These are different diseases. They don't change in the same linear way that typical development does, right? And so developing a fit for purpose scale, we did it for AGS. We did just a simple developmental inventory. If you're going to do it, make it simple. That's the key. Don't get fussy. And then repurposing. So I saw you did the Hammersmith and the AIM, and like there are a lot of scales developed more for the neonatal follow-up clinics that potentially you can repurpose, like a lot of the SMA. So we do a lot of the repurposing of SMA things for like kind of a low item function battery. But if you're expecting a huge gain in developmental skills, you're going to run into the problem of ceiling effect if your therapy is really good. And so one of the challenges with the fit for purpose low item function scales is that, like the off-the-shelf ones, is the cap. And so that's where developing your own scale that allows you to catch that like full upward trajectory might be really helpful. We can talk. And then we do validation to make sure that before anyone administers a scale, either retrospectively or prospectively, they pass the same rigor that you would have to for a clinical trial, where you have to certify that you know how to administer it appropriately. And for someone to administer a retrospective scale in our natural history studies, they have to have a minimum of 80% concordance with the prior raters. And we make them do test samples, and we make them do it over and over again until they hit the key metrics, because again, you need that rigor when you're doing those comparative trials retrospectively too.
And I would add that it's important to understand the changes in different domains of development. I think, Laura, you were saying it very nicely earlier when you were talking about the AGS kids having normal cognition, and good language and understanding, but maybe not able to express and to test in typical ways. And I think this is kind of similar to CP scales, where we might not be seeing these changes. I know in the Canavan group, we see a lot of improvements in receptive language and cognition, but they struggle with expressive language primarily, I think, because of the motor impairments that persist. So kind of thinking through those issues when you're scale developing as well.
Is anybody monitoring the Zoom? Are there any questions? Oh, they're typing again. Well, In the meantime, I have another question. So, it's been sort of as I listening to all the different talks, I hear that, I get the sense that using viral mediated therapies for gene expression and antisense oligonucleotides for gene knockdown, is there any benefit to using viral mediated therapies for gene knockdown in leukodystrophies, and maybe perhaps a comment as to what you think on the question I put here, how do these two therapies compare in terms of safety, efficacy, any of that?
Can I clarify to see if I understand? I think the comparison of knockdown versus gene replacement strategies.
Yes, they're using the shRNAs, and I did a search and I didn't see any of those.
Yeah, I mean, they're completely different approaches and they won't work for all diseases. So you have to make sure that you're matching the right intervention to the challenge that you have in the cell. And so I think cell targeting is a major issue too. And so not only do you have to be able to fix it in iPS cells in the lab and in mice, you have to be able to get the therapy into the right zone. And so a lot of times when we're choosing either knockdown or gene replacement, it just has to do with the general genetic mechanism, but also the cellular mechanism of it. So I would say overall, the siRNA therapies are really safe, but they're not effective in the vast majority of our diseases. And so to me, that's the guiding principle is, can I fix the defect in the right cells in a safe way as possible?
Josh.
I guess I was going to add two comments about the kind of safety profiles. One is that something our neuromuscular colleagues have dealt with, and I think now we as a field in leukodystrophies are catching up in their footsteps, are that we're seeing complications of ASOs and gene therapies and other kind of targeted therapies that are kind of class effects. So they're not specific to the disease necessarily, but they're specific to the class of therapy. And I think that we as specialists doing these therapy trials know those problems, but our colleagues, even in our own divisions, our own departments, don't know those. And so I think we have a kind of a education type issue we need to be thinking about for either our trainees or for our institutions, how to set up safety mechanisms to take care of a patient who has a complication of a therapy. So I think that's something that neuromuscular diseases are seeing, and that I think now we're starting to see. The second point is that that I think in the neuromuscular world, I've now heard several people say that, oh yeah, they've treated someone with the gene therapy and then they uncovered some second hit mutation that the kid had because the kid got diagnosed for the primary phenotype with like a specific gene panel or something like that, but hadn't done a more exhaustive genome or exome to identify that they had a second hit. So I think that we probably should also be thinking about anyone who goes on to a multi-million-dollar therapy or clinical trial should probably have a exome or genome level test.
I would also add that I don't think it's either or. I don't think that, for diseases and disease types and biologies that are amenable to multiple approaches, right now for most of our disorders, we feel like one approved therapy would be a massive win, and it would be. But I think, again, looking towards neuromuscular and SMA, they found a lot of success in combinations of therapies. So it's not just gene therapy and you're done, or an ASO, then you're done, maybe in a gene therapy and an ASO or some combination of these approaches would actually lead to the best outcomes.
We have a question here.
For bringing the safety issue up, I want to bring that back to ask how all of you as PIs would navigate situations where these companies close down, and then you've recognized either positives or negatives in that trial. What is our obligation as PIs in those situations to disseminate that knowledge, and how do you navigate, the fact that you have signed a non-disclosure with the company, but you also want to educate your colleagues on it?
The microphone's in front of me, so I'll talk, but someone else jump in. I mean, Deepa, I think that's a brilliant point. I think we've been burned so many times in rare diseases with companies shutting down prematurely. I think we should figure out ways in the contracts to get it built in so that the non-disclosure and the siloing of information ends when the trial ends, so that the information can be disseminated. I think there's also a lot of movement to try and set up more central repositories for safety tracking across the gene therapies, because we don't know. It's all siloed in clinical trials for the vast majority of things. And so you don't know these safety events, and you don't know what's common, as to Josh's point, what's common across different trials that might be coded in different ways or not released for years and years. I think it's going to be a huge barrier.
I think it's one thing if it's like you're not able to report on like efficacy, whether it was or was not efficacious, but when there is like safety issues that are going to affect the other patients that have been dosed, I think it's unethical for the PIs just to be, you know, I mean, you have tape over your mouth and you're not able to talk because of a legally binding document, but that's legally binding. And so you have to uphold it. But I totally agree. There needs to be some way for at very minimum safety data from a clinical trial to be able to be shared, because these companies have way too much power in that, because they just shut the door and say, okay, what's hidden behind the door is hidden behind the door. But it's really not fair to the patients and the field as a whole to know what could be coming down the pipe for them.
You know, this is not like, oh, this company has this drug and it didn't work, it shut down, but you know, it's a class effect. What could we learn from that, that we could disseminate for the future?
That's common. There's just actually, as of last week, there's some good things happening for once with the FDA. The commissioner requested release of communication that goes back and forth with all companies that has to be publicly available. So that was just last week, and there's a website. I'm gonna find it. I can't remember on top of my head what it was, but there's a place now to go to where you can see the communication between the sponsor and the FDA. And so, you know, obviously they are obligated to report it to the FDA, even though it's proprietary information, but it no longer will be. Now, I don't know if there is going to be an embargo or not. That wasn't clear with that announcement. And there's only a file that was dumped online right now that includes, you know, I think 200 something reports. I looked up all the leukodystrophy names that I could think of, and none of them were in there. So, but that information will actually be publicly available, which is great.
I think the other thing that really makes me nervous, Allison, is the long-term follow-up needs too. What do I do if we have dosed 5 children and the study shuts down? I don't personally have money to pay for the safety monitoring, right? Let alone the efficacy monitoring. And so I think making sure we set up the safeguards that if you enroll a child, you get the money designated to care for them, right? I would rather enroll less children in a trial safely than enroll more children and have my feet cut off a year into it. So I think those are the things that, as we're realizing the churn of these rare disease companies is so great, I think we just need to be really savvy about protecting ourselves and protecting the patients.